I'm here tonight with you because of this man. This is my father, and of course, I owe my existence to my father, but I also owe him the reason. He is the reason I'm here tonight on this stage. My father was a glider pilot in World War II. He came home, he married, he had 3 children. He got his aeronautical engineering degree from Cornell, and in fact, my father was the chief test conductor of the Atlas. That's the Atlas there. It was America's first intercontinental ballistic missile. At the age of 39, my father got a diagnosis of lung cancer. He died several months later. Cancer runs in my family. My mother beat breast cancer twice. My brother Tom was diagnosed with non-Hodgkin's lymphoma when he was 32 years old, but he beat it. He lived another 27 years until he received an even more dire cancer diagnosis of glioblastoma, of brain cancer. I was in Washington, D.C. working at the time of the diagnosis. I left my job, I came home to Albuquerque, New Mexico, and along with my brother Tim and Tom's lovely wife, Madeline, we took care of Tom. And he was amazing. He was so conscious of being given 27 more years. He would say to me, honestly, I'm the luckiest man alive. And I would say to him, I wouldn't go that far. It was at this time that I came across this. I finally got the slide right, right? This is a picture of lung cancer cells that have been labeled with a porphyrin. Now, porphyrins can be organic, like the heme and hemoglobin that gives you that bright red cell, bright red color, but this is a synthetic porphyrin, and Los Alamos National Lab researchers had found that by labeling cancer with this synthetic porphyrin, they were able to detect lung cancer in slides from sputum. And we've all just eaten, so I won't give more of a demonstration than that, but that mucus that comes from our lungs. Los Alamos has transferred that technology to a startup in New Mexico that asked for my help. And we were able to, with a bit of lobbying, get funding for a clinical trial. And that clinical trial showed that labeling sputum from individuals who had cancer and labeling sputum from individuals who had, is that better? Okay. And labeling sputum from individuals at high risk, and then looking at those slides for these red fluorescing cells, that we could tell the difference with greater than 80% accuracy. So it was the science was moving forward as a diagnostic. Unfortunately, the little startup was not. It had run into some difficulties. So my brother Tim and I are attorneys. We did what attorneys do. We reorganized. We went to court. We reorganized the company. We were able to attract some very sophisticated and very experienced shareholders, investors, and together we started to look for a new home, because the one thing that we knew we needed that we didn't have was a really strong scientific team. And indeed, after looking at a half dozen cities, we selected San Antonio because we found Dr. Vivian Rebel. Dr. Rebel is a MD, PhD, a hematopoietic cancer stem cell biologist who was at UT Health, had her lab there. She was fascinated, as we were, with the technology, and we started a collaboration. Now, what that means, her background means that she is particularly expert at finding rare cells. And cancer, especially at an early stage, is a rare cell. So we went to work. We started a collaboration. She built and assembled an excellent research team, and we were able to move the technology forward and the company such that she became and is our Chief Science and Medical Officer and Executive VP, as well as the research team came to BioAffinity. So thank you, UT Health. I can't see you with the lights, but thank you very much for your remarkable researchers, those who have come to BioAffinity and those who we collaborate with still. And thank you, UTSA. That is our home. We are part of UTSA's innovation and commercialization program. And thank you, the city of San Antonio. You are a shareholder in BioAffinity, and what could be better than that? Indeed, San Antonio has become a wonderful home. Over the last 5 years, we've been able to take this technology from a slide-based system where you look down and you look for red fluorescing cells to a flow cytometric platform, a flow cytometry test that is able to look at an entire sputum sample, averaging 21 million cells in literally minutes. And with unique gating strategies and labeling, we can find those rare populations. We can look at cell populations in sputum and find biomarkers like that high red fluorescing cancer cell. So we're moving forward and finding lung cancer. And let me tell you why that's important. Lung cancer is the leading cancer killer. A diagnosis overall of breast cancer, for 5 years, you have a 90% survival rate with breast cancer. For prostate cancer, the 5-year survival rate is 98%. For lung cancer, it's 19% overall. And that's because we find lung cancer so late. But that's where our test, we call it CyPath Lung, that's where CyPath Lung can really make a difference. CyPath Lung can find cancer earlier, and it can detect it when we can treat it. Now, there is screening available, and I encourage anyone who is at high risk to be screened by low-dose CT. Screening can drop that death rate by 20%, but it comes with a high false discovery rate, which means that for every 100 people who get a positive screen, only 4 have the disease. So CyPath Lung, again, can be used in that arena to lower that false discovery rate. We have licensed the technology to Precision Pathology Services that has been providing stellar pathology services for many years in San Antonio and is licensed in 23 states. Ours, indeed, is a success story. And tonight you will hear 4 more success stories that are equal or greater than mine. And you have to ask, why? How do we do this? Why do we do this? Well, I can't tell you exactly why everyone does it. I can only tell you my story. It is that I think of my father. I think of a life so full of promise that was cut so short by so late a diagnosis. And of course, I think of my brother Tom. This is Tom. He used to always come to all my speeches. It's appropriate, he's here. I think of what he said to me. I got 27 extra years. And I know that CyPath Lung, that our test, our non-invasive test for lung cancer, will give more people more time. And so more people will hear what I heard from Tom. I'm the luckiest man or woman alive. Thank you.